Everyone, I'm Francis Collins, Director of the National Institutes of Health. It's my pleasure to be introducing our speaker today, Dr. Margaret McFall-Nguy, who is with us for the second Walls Lecture of the year. She is currently Professor of Medical Microbiology and Immunology at the University of Wisconsin in Madison. I read a little bit of some of the things that she has been writing about, and also an interview with her in the Huffington Post, which had an interesting title, "The Extra Pounds You Can't Afford to Lose." I think we'll be hearing about that today because, of course, everybody is very focused on the pounds you want to lose, but you don't want to lose your microbial pounds. It will be bad for you. And that's the topic she's bringing to us about the microbial world, and particularly the way in which this has created a new view in evolutionary terms of how animals and their microbes must co-evolve to be successful. Her background is a Bachelor's of Science in Biology at University of San Francisco, followed by PhD in Biology at UCLA, postdoctoral fellowships, including one supported by the NIH in an NRSA award, and then beginning at USC as assistant professor and moving on to full professor, and then at the University of Hawaii. That must have been fun. And then somehow she managed to leave Hawaii and go to Wisconsin. Now go figure. The transition must have happened in the summer, I'm guessing. She is PI or co-PI on current NIH grants supported by NIAID and NIGMS. So she is a grantee of this institution. And her research has focused very heavily on these symbiotic associations between animals and bacteria. She's an elected member of the American Academy of Arts and Sciences. She's had a Guggenheim Fellowship. She comes to us, I think, at a very interesting time because the amount of data that is possible to generate on the microbiome has been expanding really dramatically in the last few years with the advent of technologies and the Human Microbiome Project. So please join me in welcoming our speaker for today, who's going to talk about living in a microbial world, deciphering the molecular language of partnership.
Okay, well, first of all, I'd like to say thank you for this phenomenal opportunity to speak here at the NIH. I'm really excited, and I hope I can provide something very interesting for the crowd. So, and I'd also like to thank Susan Gottesman for being such a great host, and Dr. Collins for the wonderful introduction. So, as it's here, I'm going to be talking about living in a microbial world, and this is something that we're learning about. Is it going on and off? Yeah. Maybe it'll fix itself. We're just beginning to understand this, and as many of you know, I study an animal model system. But the other side of my life is I try to, from an evolutionary standpoint, try to get across to people the importance of understanding the history of interactions with microbes and what is going on in current day. So my talk will have two parts. One, I will talk about the fact that what's happening right now is technology has brought biology to an inflection point. And in fact, we are in a position now that we haven't been in for many, many, many decades. And that is, I think we're going to see some big changes. And it's based on the technology, molecular technology associated with sequencing. And it's creating a huge change in the way we see the entire biosphere, and mainly the relationship between macro and micro worlds. Then the reason why I'm starting here is because this particular change in our view has tremendously impacted the field of symbiosis, such that now, I started in symbiosis as a graduate student at UCLA back in the 1980s, and it was, just another small field. But now it's becoming front and center in biology. And so most animals and plants live in very close and likely coevolved associations. And so I'm going to talk a little bit about that and then give you some examples from the work that I've been doing in my lab. Just want to take you back. And, I'm sure you guys have heard this many times now, that microbes are really at the base of lots of things. I want to show you my particular viewpoint, because I'm hoping that if enough people tell this story enough different ways, that it will begin to sink in to the community. So if we go back and think about how we classified the biological world historically, it was mainly with visual analysis for most of the time. And so you start way back here, and people were trying to figure out how things related to one another way back here. And then Anton van Leeuwenhoek and his microscopes, so that's where he saw the first microorganisms. So he went from classifying the world as animals and plants to animals, plants, and microbes. And then in the 20th century, we had the advent of the TEM, and we had high resolution visual analysis of things. And that, there was this guy, Whitaker, who, based on cellular morphology, he derived something called the 5 kingdom model. And here are the 5 kingdoms. But the next thing that happened, this didn't last a whole long time, because the next thing that happened was molecular analysis. And that molecular analysis is shaking the world of biology. So if you look more closely, what went on in here, a lot of this, of course, was done by Carl Woese. And he started in the late 1970s, but technology always lags behind. And he was working in here, and it wasn't until PCR was really widely used that you could begin to do this more quickly. But in this period, he was working hard to create a phylogeny. And he finally published a paper in 1990 that convincingly showed the biologists that there were 3 domains of life: the bacteria, the eukarya, and the archaea. Now, the interesting thing is I did a sabbatical last year at Caltech, and I was talking to one of the faculty there, and he said to me, he said, well, you know, there are these 5 kingdoms of life. And this is a really smart guy. I looked at him and I said, where have you been? You know, in other words, the general population of biologists hasn't necessarily caught on to these ideas. And so our understanding or our acceptance of these ideas can lag behind. And textbooks, so I've taught introductory biology a whole bunch of times, and textbooks to this day often will show the 5 kingdom model as an option. But biology really has turned a corner in the last 5 years, and we're really beginning to get a much better handle on this, even than Carl Woese could have. And so what I'm showing here is the dollars per megabase DNA sequence. And this, of course, is Moore's law. And then in 2006, it was $6,000 a megabase, and today it's 10 cents. I mean, that is tremendously enabling. And then you can see over here how back in 2002, 2004, there were very few full genome sequences, and now there are a huge number. So this is absolutely enabling and revolutionizing the way we see the biological world. So what this meant is this guy who did the 5 kingdom model had the fungi, animals, and plants as, you know, the majority of the biosphere, and these were the microorganisms, the protista and the monera, and there were no mechanisms by which to determine their phylogenetic relationships. So we turn to the 1990s through the present, and so in most classrooms across the country and maybe across the world, Carl Woese and George Fox's 3-domain model, the bacteria, archaea, and eukarya is what is taught as the organization of the biological world. So it's called the 3-domain model. Now this, because of all that sequencing that's been done in this last 6 years or so, this is by no means not controversial. And so if you looked at some of the things that are going on here, Eugene Koonin here at the NIH is arguing that there may not even, because of rampant horizontal gene transfer between the bacteria, the eukarya, and the, not so much the eukarya, but the bacteria particularly, and bacteria archaea, that there may really be no robust tree of life as we think of it. And that's not to say that there's no evolution. There's certainly selection pressure. But I mean, this is just amazing. There are other groups, the protistologists, the animals and plants are sprinkled around here. They think that the protists are at the base of everything. And then over here, there are some people that are feeling or are positing that there is a fourth kingdom with the divergence of the large DNA viruses. So I guess the point is here that our idea of the organization and relationships in the biological world is drastically changing on a yearly basis. And it's happening, and it's like there was a book written in the early 1970s by Alvin Toffler called Future Shock. And I know most of you in this room are probably too young to remember that book. But that book was all about too much information in too short a time. And that's pretty much what's happening to biologists at this moment. So now one of the things that when you start thinking about the diversity of the microbial world, and you see that microbes are the most abundant organisms, when you then look at their metabolic scope, it's also very impressive. So the eukarya have a limited metabolic diversity. So the eukarya here have limited numbers of terminal electron acceptors and electron donors. It's amazing, with the origin of the mitochondria, or the uptake of the mitochondria, which was thought to be a seminal event for the radiation of the eukarya, there was thought to be one single event that did that, which had to be an incredible adaptive advantage. Of course, oxygen as a terminal electron acceptor is the top of the electron tower, and so you get more bang for your buck from that, and that's probably why it was such an advantageous thing. But imagine that canalized that entire group of organisms into a single terminal electron acceptor for aerobic respiration. So that's pretty amazing. When you consider that the bacteria, the archaea have sort of an intermediate diversity, but the bacteria, look at the list of electron donors and electron acceptors. Tremendous diversity. And so when various biologists talk about humans and mammals and whatnot being complex, I think the microbiologists can make a pretty good argument that there are microbes, certain microbial species can have numbers of these electron donors and acceptors. So we've got the eukarya, which are limited in metabolic diversity, and we've got the bacteria, which are very diverse in in metabolism. So looking at a few more of the traits of microbes, microbes, of course, are relatively small, usually. They have short generation times, relatively. They have large population sizes. They have a propensity, as Eugene Koonin is showing us big time, for horizontal gene transfer. The pangenome in some of this is thought to be limitless, and they're tremendously metabolically versatile, like I just mentioned, On the other hand, let's look up at the animals and plants. Multicellular organisms, of course, relatively speaking, tend to be large, have long generation times, small populations. Propensity against horizontal gene transfer, and why that is, we're not exactly sure. And they're metabolically limited. So by partnering together in a symbiotic association, they drastically increase one another's scope. So why not make partnerships, long-lasting, persistent, coevolved partnerships? If you can take advantage, each side can take advantage of the other's major traits. So the fallout of the genomics revolution that I was talking about, when you think about these traits, is the recognition of symbiosis as a very general phenomenon in animals and plants. So in plants, for instance, they've just found that about 30% of the carbon that is fixed by a plant is diverted through its roots into the surrounding soil, so that its microorganisms will be fed and structured to that specific plant. So the soil, or the rhizosphere, is like the plant's gut, essentially. But 30%, that's a huge layout to manage your microbial partners. In humans, as you guys all know, there's this sort of hackneyed set of facts down here that we're 90% non-human. If you count the mitochondria, we're 1 to 200 about. We're 1 to 1 by gene number, and by gene diversity, the microbes are about 200 times more gene diversity. We are covered, Julie Segre and various other people here study the various sites in which the microorganisms find themselves, but components of 8 of the 10, so vertebrates are characterized by 10 organ systems. That's their body plan. And 8 of the 10 vertebrate organ systems have components with coevolved beneficial microbes. So this is a big deal. And so you know that humans have now, or do now, and have throughout their evolutionary history been influenced by interactions with microorganisms. And this excuse me, diagram I'm showing here of the various impacts that people have shown, are beginning to show to the immune system, to the circulatory system, and so on. The microorganisms seem to be impacting just about every aspect of our biology. And all these data are within the last 3 to 4 years. I mean, it's just exploding. One of the most interesting papers that I've seen recently is the interplay between the microbiota and the brain. So you know that the microbes are not in the brain. It's just that they're very metabolically active. The molecules that of their metabolism are taken up by the intestine into the circulatory system, and they affect do now and have over human evolutionary history affected every cell that is serviced by blood, and even those that aren't. I mean, they can deliver things across the blood-brain barrier. So, I mean, this is phenomenally deep and something that people should consider. So what's the problem? Okay, in my mind, we have a severe conceptual challenge to deal with. And that is that biologists, microbiologists particularly, are very influenced by a conceptual framework that began in the 19th century. And that conceptual framework is that microbes, as organisms, are either pathogenic or they're out there. So they either make things sick or they're out there in the soil or the seawater or something like that. And I can tell you, when you go to the ASM meeting, that the American Society for Microbiology meetings, that meeting is divided into people that study microbes for understanding basic molecular mechanisms, but when considering microbes as organisms, they consider them as pathogenic or they consider them as out there. Okay, so what is the conceptual home for thinking of the body of an animal or a plant as the natural home or the natural evolutionary state? There really is no conceptual home. So this is a big problem because we're siloed into departments and various institutes based on this particular set of prejudices. Okay, so thinking about how we approach this and what kinds of experimental plans we're going to develop. If you look at what is highly conserved among the symbiotic associations, you can make a short list. So widespread evolutionarily conserved features of many, many, many symbiotic associations is that they're extracellular. They often occur, or very often occur, along the apical surfaces of epithelia. So since the dawn of animals, there have been bacteria associating with the apical surfaces of epithelia. They're usually horizontally acquired. There are whole groups of organisms that vertically acquire them, or that is in or on the egg. But usually, each generation, the bacteria are picked up from the environment, an environmental reservoir. And then in the vertebrates, they are highly complex. And so I would argue that there are a lot of invertebrate situations in which the complexity is very low. So this is sort of the conserved set, in other words, what most animals do. What are the major questions we want to ask? So we want to ask who's there, and there's a huge, huge effort going into who's there. But there's also a series of other questions that relate to these features. Firstly, how in the world do you get them, especially if they're a rare organism in the environment? How do you get them? In the water to land transition, when animals moved onto land, many times what they did was they have mechanisms by which to ensure transfer. So a cow, for instance, will lick her udders, and in that way pass the microbiota of the rumen to the calf. But there have to be this horizontal transmission. With horizontal transmission, there have to be mechanisms by which to pass and acquire each generation with fidelity. And it has to be the right subset. So you don't just associate with some random set of the environment. It's a very specific recognition system that's coevolved. And development, they have to come together, and each of them affects the other's development. And then finally they become stable. And that stability is a very critical thing. You don't want the host to either be overgrown by their symbiotic partners, nor do you want the host immune system to get rid of the symbiotic partners. So there has to be some sort of détente established at that point. And then the very last thing is, what in the world is the difference between mutualism and pathogenesis? I mean, that's a huge question. I mean, we're covered with microbes. And now we know that most of our interactions with microbes are beneficial. On occasion, we have a pathogen coming in. And the pathogen doesn't come in to the animal's body, it comes into an ecosystem. And when what is happening when that pathogen comes into an ecosystem? So I'm going to tell you about using animal models to try to drive a wedge in some of these questions and the value of using these. So what I've done here is I've taken a slide from a review that Ned Ruby did for Nature Reviews Microbiology. And I should say that this list was taken from the Nobel Prize Award, the Nobel Committee website. And so what I have here is the list of recipients of Nobel Prizes, the discovery recognized in the models. And you'll see that in the field of developmental biology, so these are all developmental biology. In the field of developmental biology, much of our understanding of how cells work and how the very basic principles of cell fate and cell division and fertilization and so forth were derived by the use of animal models. And so they've been incredibly beneficial. Now, I think, and I would think this, I think that because with developmental biology, it's kind of interesting. There are something like 38 animal body plans. There are 38 phyla. And so development actually really spreads out, and it actually creates a whole bunch of different things. The way that eukaryotic cells have interacted with microorganisms is as old as the hills. And I would suspect, and you will see in what I talk about, that the language is very, very old and very, very highly conserved. And one would expect that if they've been, living together for such a long time. But what I'm showing here is I'm showing a set of experimental models of the persistent colonization of epithelia that are really have been under, pretty strong development. And so in the vertebrates, I call this kind of an engineered model. And the reason is because these are not naturally occurring organisms. They're laboratory strains, inbred strains of mice or inbred strains of zebrafish. And they're gnotobiotic vertebrates. I need to say right up front, in no system, no animal system, is there genetics in all partners. No animal system is there genetics in all partners. So in the plants, in the Rhizobium leguminous plant symbiosis, they have genetics in both partners. In no animal system do you have genetics in both partners, in all partners. That's going to be really tough here. You can have genetics in the mouse, you can have genetics in the zebrafish, but to get genetics in all of the partners and to try to understand what's going on there will be difficult. One of the things that we can turn to for some heuristic value are the invertebrates that have a range, and oftentimes they have simple consortia in their guts. So for instance, what people are finding with Drosophila is that they have something like, 5 or so phylotypes in their gut. And so they have a very simple system. And there are other models that are undergoing development which are really, really exciting. But one of the things that's fun about this is that you have such few numbers that naturally occur that you can hope to make mutants in some of those microbes and see how communities are structured. Some very basic rules about how communities are structured. And then finally down here are the binary associations. Now obviously not many animals have binary associations, so that in itself makes it unique. But it gives us an opportunity to really look carefully at how microorganisms talk to host cells. So what it's like is it's like the difference between being in a really noisy cocktail party and being in a very intimate conversation with one person. In other words, you're able to decipher the context and the content of that much more easily with a one-to-one conversation. And so the nematode vesicle, the earthworm nephridia that Dave Stahl's doing, this is Heidi Goodrich Blair and other people. And then I'm going to tell you a couple of short stories about how the squid has shown us some work. So this is the animal that I work on. So the squid vibrio light organ symbiosis, over the last 22, 23 years, we've been developing as a model for the study of the chronic colonization of animal epithelia. And so what you see here is the Hawaiian bobtail squid. And this is not a giant's hand. This is my technician's hand. So it gives you an idea of how large these animals are as full adults. They're very small. They're great laboratory animals. If you were to open this animal, flay it open, you would see that it has a complex bilobed organ in the center of the mantle cavity. And deep inside, there's an epithelium that supports a pure culture of the luminous bacterium Vibrio fischeri along the apical surfaces of a highly polarized epithelium. So what this animal does, I always forget this because I'm not a behaviorist, so I was sure to put this slide in because people always want to know what they're doing with the light of the animal, of the bacteria. So what the animal does, it's a night-active predator in the shallow sand flats of the Hawaiian archipelago. It comes out at night and it hangs above the surface, above the surface of the seafloor, looking for things to eat. And it emits ventral light from the bacteria out of its bottom side, matching downwelling moonlight and starlight so that it doesn't cast a shadow against the visual field of a predator looking up from below. So it's a camouflage. It's a Klingon cloaking device or something. This animal has a way to camouflage itself using bacterial luminescence. And if the moon goes behind a cloud or whatever, it has the ability to make it dimmer or brighter by pulling back. It's embedded in the ink sac, and the diverticular ink sac move back and forth. Also, the luminescent reaction is oxygen requiring, and the animal withholds oxygen from the bacteria. So not only did I move from Hawaii, but the squid on a regular basis make this kind of rude trip from Oahu to Madison, Wisconsin, where we maintain them in aquaria there. You only need maybe 10 females and 6 males in the lab, and they will keep you in 50 to 100,000 juveniles a year. So they lay eggs, the females lay eggs on hard substrates, and any given female will lay, 15 clutches of 200 to 300 eggs. So it's really an easy thing. Here's a clutch of eggs. Each one of these is about a millimeter. And the babies hatch. There aren't any larval stages, and they are ready to be colonized right away. So the major questions that I mentioned to you, we've been really lucky because the system has lent itself to looking at each and every one of these and other sorts of questions like the ecology and evolution of these systems, but in how they work sort of in that conversation. So one of the things we've done is we found that MAMPs, or microbe-associated molecular patterns, specifically lipopolysaccharide and peptidoglycan, actually induce development of the squid, and the pattern recognition receptors actually are involved, of course, as receptors of those MAMP signals. What's cool about this is that the squid, we published a paper on this in 2004. I'm over at the Pasteur Institute, and I had been a speaker, an early speaker in the meeting, and I sat down, and the guy who got up after me was still showing a squid slide. And actually, he works on the mouse at the Pasteur. And it's always been known that gram-negative bacteria-induced development of the gut-associated lymphoid tissue, and nobody ever knew why. And so he said, hey, if a squid can do it, maybe a mouse can do it. And in fact, the exact same molecules that that induce development in the squid induce development in the mouse. So the other thing that we found is we found a very complex diel rhythm to the issue of stability, and this is what I'm going to be talking about tomorrow in Lambda Lunch, is the complex circadian rhythm. Now what's interesting, in the mouse, they found a complex circadian rhythm on the epithelium with cry and timeless and pure and all the clock genes expressed, on the epithelium, on the mucosal immune system of the gut. But what they're missing is they have not determined whether or not the bacteria participate, are either affected by or actually entrain the circadian rhythms of the host. And in the case that I'm going to talk about tomorrow, the bacteria are absolutely essential. And so I think that's something that will be forthcoming. And then lastly, in mutualism versus pathogenesis, we're finding that it's the exact same language. Every single time we find something, it's largely what goes on in pathogenesis. So what it makes one think is that because pathogenesis is now known to be very much less common than beneficial associations, maybe we shouldn't be calling these virulence factors and host defenses. Because in fact, they are the language whereby an animal talks to its normal microbiota, and pathogens might be thought of as spies in a pre-existing conversation that the host has with its normal microbiota. But I'm going to go into depth now in just a couple of short stories. And I'm going to talk about how in the world do they get their symbionts, and how do they know it's the right one. Engaging Fisheri and only Fisheri. So basically what happens is they make a habitat transition. Vibrio Fisheri is a member of the bacterial plankton. It's out there and free in the seawater, and then it colonizes the crypt spaces of the animal. And what you've got here is you've got a GFP-labeled Vibrio Fisheri and an RFP-labeled Vibrio Fisheri that you can see through the body wall of the animal. So it's going from about 5,000 cells per mil against a background of 1 million other environmental bacteria per mil to being at about 10 to the 11th bacteria per mil of a single species. So that's a pretty darn good habitat transition. It happens all within the space of about 8 hours. So before I get started, I want to give the acknowledgements. The studies I'm going to talk about were done by Spencer Nyholm, Shana Davidson, Melissa Alter, and Natasha Kramer, terrific students in my lab. And in Ned Ruby's lab, my partner who works on the microbial work, does the microbial work, Yanling Wang, and Caitlin Brennan with funding from NIAID, NCRR, and the W.M. Keck Foundation. Okay, let's get started. So the juvenile organ is different from the adult organ in that it has two ciliated fields on either side, and it pulls water in, and all the environmental water containing Vibrio fischeri passes, or a lot of it passes over the organ. And it has this, if you were to look closely at this, you would see that there are these two ciliated fields that are an epithelium with a blood sinus and 3 pores. Those 3 pores on each side lead to 3 independent crypts where Vibrio fischeri will colonize. The entire process can be observed in real time. You can sit there and watch this on the confocal. So over 3 hours, Vibrio fischeri cells gather outside. So this is the light organ labeled with rhodamine phalloidin. And you can see these are GFP labeled Vibrio fischeri that are above the pores. This is a huge inoculum so that we could see these big aggregates. And then at some signal, and I'm going to tell you what that signal is, they begin to dance into these pores and colonize host tissues. But they wait and wait and wait, and then they go. So after aggregation, the symbionts migrate down 3 independent ducts. And so I'm going to show you what that looks like by confocal. And so you're seeing the cilia on the surface, and that's where Vibrio fischeri is going to interact. And so, is Anton here? I'm just going to ask for the lights down just a little bit. There's a duct here, and a duct here, and an antechamber, and a bottleneck. And so you can see this long journey. It's not that long. It's actually about 50 to 100 microns. But that's the gauntlet that Vibrio fischeri will go to. And actually, the crypt spaces are out here. They don't. This is tubulin staining, and they don't stain with tubulin. So only the ducts stain. So the symbionts, as I mentioned, are extracellular, and they proliferate in the crypt spaces. So this histological cross-section gives you perhaps a little better idea. So here's a pore. They go up this long duct, which I'm going to show you as a gauntlet. They interact. They come up here and proliferate, interacting with 2 cell types, the epithelium, and some hemocytes that do the diapedesis. They come into the space, go back into the general circulation, and tell the animal about the status of what's going on in there. That's what we think. The colonization, I should mention for sure, is signaling, colonization of and signaling by only occurs by Vibrio fischeri in the light organ. So it's restricted to Vibrio fischeri here. So one of the things that's interesting is despite the fact that you might have a big aggregate of cells, you only have a single cell, as though there's competitive exclusion. Although there's competitive exclusion on the system. So these are GFP labeled and RFP labeled cells. And so each one of the crypts, there are 3 crypts that are labeled here. And each one of the crypts has got either a green cell or a red cell. It sort of goes to the issue of how do you make sure that you've got a good strain in there. So it looks like there's competitive exclusion. So Vibrio fischeri in this process has 2 big challenges: to become exclusive, as I mentioned. So I'm showing here just how exclusive the situation is. So in a background of 10 to the 6th other environmental bacteria, if you give Vibrio fischeri and Vibrio perihemolyticus its sister species, in a 1-to-1 mix, you'll see that after about 3 hours, Vibrio fischeri is the dominant, even if it's a motility mutant. It is the dominant in the aggregate. And you can change the fluorochromes, it doesn't matter. But tremendously exclusive. And they are not growing there. Fischeri is not growing. It's being harvested. It's being gathered. And so they become exclusive by some mechanism. So once again, they're only about 0.1% of the bacteria plankton. And within 3 hours, Everything else is winnowed away. Then they have to find their way. And how do they do that? And they do that, like I said, they gather, and then at about 3 to 4 hours at some signal, they go in. Now, one of the things I wanted to mention is I'm going to show you a lot of things where you have a lot of cells gathering. But under normal conditions, on average, only about 5,000 Vibrio fischeri per mil are in the seawater. And under those conditions, you get a small aggregate of about 5 cells. And so you get this little tiny set of cells. And I find it remarkable. I mean, one of the things I'm going to tell you is that only 5 cells will cause a global change in gene expression across the entire organ that you can pick up in an RNA-seq experiment. Only 5 cells. They know their partner, and that is against a background of 1 million other cells. So that's pretty phenomenal. So under natural conditions, 4 to 5 cells attach to the cilia on the surface, aggregate, and a capsule is required for aggregation, and aggregation, that is, they form a tight aggregate, is required for colonization. So how does it work? How does this work? Well, one of the things I wanted to mention is that I've got this really fun collaboration going with the physicists at Caltech and USC, the engineers and mathematicians and so on. And John DeBerry's lab with his graduates, his actually now postdoc, Jana Nauroth, and Eva Conso is a mathematician, and Scott Fraser is an imager. And what we're doing is we are going to be looking at the physics of how these ciliated fields work. And what's really exciting about it is we can watch a natural system with how bacteria interact with a ciliated field under natural conditions and how they select a specific bacterial type. So we're really excited about this. And we'll be doing all kinds of mathematical modeling and whatnot. But that's in the future. I want to tell you today about what we've done as far as the chemistry is concerned. So at 60 minutes, the very first thing that happens is you get just a huge amount of mucus shedding. And so you've got all this mucus shed here. And the mucus degrades, the mucus biochemistry dissuades all of the gram-positive bacteria. So the first thing that happens is the gram-positive bacteria are dissuaded and are gone from the situation. So there's the first winnowing. And they do this because in this mucus is lysozyme and peptidoglycan recognition protein, which break down the cell wall of gram-positive bacteria. But the gram-negatives are still around. They seem, in the absence of fischeri, they'll make beautiful aggregates, but they go to the pore duct interface and they're dead. So something else is going on. One candidate is host nitric oxide. So this is a really fun story. So by immunocytochemistry, you see that there's nitric oxide up in the arms, a little bit of nitric oxide, but the ducts are on fire with nitric oxide. So the bacteria face this tremendous nitric oxide gauntlet, and this is just staining the nitric oxide. This is the antibody, and this is the fluorochrome that sees nitric oxide. If you inhibit nitric oxide production, you allow nonspecific bacteria into the host tissues. So one thing that's really wild is that host nitric oxide is released out into that mucus. So the nitric oxide that's in the arms is released out through the cilia into that mucus where the bacteria are aggregating. Now, Vibrio fischeri has genes that mediate response to nitric oxide. So they have a way to respond to what the host is saying. They have an HMP, which is a gene that encodes a flavohemoglobin that detoxifies the nitric oxide to nitrous oxide or nitrate. They also have another gene called HNOX that encodes a heme-containing soluble guanylate cyclase that binds NO and then causes changes in gene expression. How does this work? Turns out that the nitric oxide that's going out into the ciliated field is priming Vibrio fischeri with just a little bit of nitric oxide, sublethal levels of nitric oxide. So if you take, what we did was we put M. cherry as a constitutive promoter, there so that you could know where the bacteria were. They were fluorescing M. cherry, and then any GFP fluorescence would be for the upregulation of the HMP. If you look at in seawater, if you add nitric oxide donors to seawater, you cause the upregulation of the HMP gene in that aggregate. Also, so that would be kind of in vitro. If you look in vivo, in the aggregate, HMP is way high, so that's here. You can add a nitric oxide inhibitor and you can turn it down. So that's one thing that the bacteria do to respond. What does HNOx do? HNOx has a heme, binds nitric oxide. It turns off this phosphorelay system, and the upshot of that is it represses genes associated with iron uptake and response to stress. So it causes the induction of genes in response to stress and turns down iron acquisition. And so it's responding to the nitric oxide stress. So how do they work in colonization with the mutants? What happens with the mutants? With HMP never competes well if it's one-to-one with the bacteria, with the wild type. And when it's by itself, it never colonizes quite well and it never catches up to the wild type levels. HNOx is really interesting. HNOx gets in there way better than wild type. And perhaps, what we're thinking is that perhaps because it is not repressed in iron uptake, it can go into the host, really grow like gangbusters. But then, with all the oxidative and nitrosylated stress in there, perhaps it undergoes all kinds of problems because it is down by 48 hours. It can't compete with the wild type later on. So perhaps all of the Fenton reactions and other oxidative and nitrosylative steps are compromising its ability to behave. So one of the things that's really fun is that the symbiont MAMPs, those things, the microbe-associated molecular patterns, specifically LPS and TCT, once Vibrio fischeri is in those crypt spaces, it tells the symbiont to stop yelling. It turns down the nitric oxide synthase production. So here are the pores of a non-symbiotic animal, and here are the pores of a symbiotic animal. And here is the nitric oxide itself. So there's a diminution. So the bacteria actually turn it down. And it's under the induction of these MAMPs. What's fun about that is that in pathogenesis, the presence of MAMPs of the pathogen almost always causes a turn-up of nitric oxide. So this is an example of using the same language but having a different effect. There's something about the symbiont that in the environment that creates a different story. So other candidates, what we're doing, got a list of people down here. We're finalists for a grant from the Moore Foundation to do single-celled transcriptomics on the bacteria. You could pull these bacteria out of here. You could look at bacteria in the plankton and look at how they make this habitat transition. And are each one of these cells saying the same thing? And as they mature through the 3-hour process, how do they change in their gene expression in this habitat transition? So we're really excited about that, and we think that that may bring other candidates that we never would have dreamed of. So not only can fischeri adapt, but its presence, the presence of only 5 Vibrio fischeri against a background of 1 million other cells, changes the game at the molecular level. So I had a student who did an RNA-seq experiment at 3 hours, exposed to fischeri and not exposed to fischeri. And robustly, we see 82 genes downregulated and 44 upregulated. It's just remarkable that that animal can tell that those 5 cells are there. So one example, I'm just going to not bother you with all of these. I'm just going to give you one example to finish up. Vibrio fischeri cells, as I mentioned, have these two challenges. And one of the challenges is to find their way. They have to figure out where they're going to go. And so they, like I said, they sit in this aggregate, and then after a while they get a signal. So Vibrio fischeri appears to chemotax. So, I mean, this looks like classic chemotaxis, right? And they appear to chemotax into host cells. So one ought to be able to put in the surrounding seawater chemicals that you might suspect would be a chemotactic agent and swamp out the gradient and see what happens. So there, if you put in nothing, obviously, so this is the colonization efficiency of a squid. When you add things to the seawater, if you add nothing, we put it just below maximum so that we can see things. These are things that in vitro Vibrio fischeri in capillary assays will chemotax toward. And so mannose, thymine, and N-acetylglucosamine, you get no response. Chitobiose, the dimer, this is the monomer of chitin, and chitobiose is the dimer. If you put chitobiose, they don't know where to go. So there's chitobiose is a very good candidate for something that might be produced by the animal. Now, in vitro assays, you can show that the response to chitobiose needs priming. This is another circumstance where the bacterium must be primed in order to respond to chitobiose. So how is it primed? The mucus matrix contains polymeric chitin. In other words, long chains of chitin. And so when Vibrio fischeri comes in, what happens is you get a minuet that primes the chitobiose. The Vibrio fischeri come in. They associate with the host ciliated field. They bind to the field. And they cause a huge upregulation of a chitinase gene. Okay, with the upregulation of that chitinase gene, and where that chitinase gene is where the protein is laid down is in the apical surfaces of the epithelia. And so it's produced and stored in the apical surfaces and then exported into that mucus. So the chitinase is exported into that mucus. But when you stain with various fluorochromes the whole landscape, you see that the ducts are on fire. That the ducts have huge amounts of this chitinase. And there's a huge amount of chitin in there. And of course, the chitinase reduces the polymeric chitin to chitobiose. So basically what you've done is you've created, by producing low levels of chitinase out here, and you've got high levels of chitinase here, you're producing a gradient of chitobiose for the bacteria to follow. If you put anti-chitinase, if you take the antibody and you tie up the chitinase in, you don't get colonization. So this percent of animals colonized. So this is without the antibody in the environment, and this is with the antibody. So chitinase activity appears to produce a gradient of chitin that the bacteria, and so that before Vibrio fischeri attaches, there's no gradient created. They attach, they change the game in such a way that they will know where to go. So you have a 3-hour process, this short 3-hour process. In fact, it's a 2-hour process, occurring over the space of about 100 microns. And what happens first is that at about 30 minutes, the host secretes mucus with cell wall degrading enzymes and nitric oxide so that all the gram-positives are dissuaded. The next thing that happens is at 1 to 2 hours, Vibrio fischeri attaches to the host cilia. Chitinase production is upregulated, and other antimicrobials are upregulated, something I didn't talk about today. There are a bunch of other antimicrobials that dissuade the other gram-negative bacteria. At 2 to 3 hours, Vibrio fischeri has adapted to the nitric oxide stress that's going to be presented in the ducts, and the chitinase activity has developed a chitobiose gradient. This is the point at which other gram-negatives are lost, and you get an aggregate that is completely 100% Vibrio fischeri. and then Vibrio fischeri at about 3 hours chemotaxis into the organ. So now stepping back, what are we really ultimately trying to achieve? We're trying to decipher the rules by which animal-bacterial interactions occur along ciliated surfaces. There are ciliated surfaces in many of the interactions of animals with bacteria with humans. And I've shown a couple of pathogens up here, Pseudomonas aeruginosa and Bordetella pertussis. And it's really interesting when you look at the scenery up close, it's obviously very similar with the mucociliary interactions. And my firm belief is that what is evolutionarily conserved is likely to be critical for the biology. And so that something that you would find as a rule in the squid, using very common virulence, MAMPs and PRRs and that sort of thing, very common virulence determinants that are conserved throughout evolutionary history, these are the sorts of things that are likely to be shared across the animal kingdom. So I hope I've been able to convince you that biology is in a technology-enabled revolution. There is, in my mind, absolutely no question. I think the question that we all have is, how in the world are we going to cope with this information? One of the major problems we have is that in universities, biology has become so siloed. There are departments that are, the department structure of universities does not favor addressing this new revolution. And also the way, even NIH, the way NIH is structured, we're going to have, it seems to me, it's going to be a challenge to try to figure out what we're going to do to recognize this. But I think this is a looming reality. So with that, I think I'll finish and take any questions.
Go to the microphones to ask questions, and in the meantime, I will, if you want to hear more about the circadian rhythms, then come tomorrow to Lambda Lunch, which is Building 37 at 11 a.m. If you need more information, just find me or some other microbe type at the reception.
Yes, Gigi?
Yeah, I wanted to go back to that exquisite selectivity that you see. Sort of how do you generate that? A lot of bacteria have defenses against nitric oxide, HMP. How are you getting such exquisite selectivity?
Gigi,
or how are you thinking about that?
There is absolutely no question that this is like a spy versus spy. Are you the right guy? Yes, I'm the right guy. Are you sure you're the right guy? Yes. There's this language that goes on. And I only presented a couple of the things we know about. Another example is pH. So out in the mucus that's distant, the pH is about 6.5 to 6.8. When you get down to the ducts, you're way down at about 5.5. So there are all these things that it seems that Vibrio fischeri, that's just another example, but there are a whole bunch of different things that we think are involved in lock and key along the way. What is true is that in the natural situation, while gram positives will not aggregate, all gram negatives will aggregate. And so, or many of them. Some of them have loose aggregates. In other words, they likely don't have the same capsule. What happens at the pore duct interface, that duct is full of antimicrobials of all kinds. Halide peroxidase, the oxidative stress in there is, through the roof, nitrosylative stress, everything. That gauntlet, so they go up that gauntlet and get to the other end, and there's an antechamber, and then they squeeze through a bottleneck. And only Vibrio fischeri can do from the pore duct interface to the end. But I think there are going to be many, many, many different things. Yeah.
I guess that idea would be interesting to test with different sort of mixes of bacteria, because it must be very subtle differences, because if you compare Vibrio with other related bacteria, I think many of the defenses are not that different.
Yeah. So one of the things that has been done is there's a Vibrio fischeri that colonizes a fish. And then we have the Vibrio fischeri that colonizes the squid. The Vibrio fischeri that colonizes the fish does not colonize the squid very well. So Mark Mandel, one of Ned's postdocs, did full genome sequence of those. And they're tremendously synchronous and very few differences. But the fish symbiont does not have a capsule, the gene that regulates capsule production. When you take that gene from the squid symbiont and you put it into the fish symbiont can colonize. So one of the things that's really fun about this system is that there are Euprymna species. So if you take the Hawaiian squid and you put it side by the isolate from the Hawaiian squid, and you put it with the isolate from the Japanese squid, and you ask the Hawaiian squid which one it likes, it will always, if they're by themselves, it will take both of them in equally. If you put them 50-50 together, the host takes them in, and then within 48 hours, it only has the Hawaiian strain remaining. So it's exquisitely specific. And so you can, with this system, and evolutionary biologists are doing this, you can look at the incremental evolution that drives specificity.
I guess I have a more basic question. What's the rate of mutation of the Vibrio in the lab, and how do you avoid a situation where the lab creates evolutionary pressures that would not be seen in the wild?
Yeah. We all work with frozen stocks. That's the only thing that I can say. We do a lot of very natural experiments. In other words, the females vent 95% of their culture every day at dawn. Many of our experiments were done with a strain called ES114. And what you're talking about did happen once. And it happened with a glycerol mutation that we didn't catch right away. But we haven't seen any, by using frozen stocks, we haven't seen any problems or any changes in ES114 over the 20 years as frozen stocks. One of the interesting things is so luminescence is 5% of the metabolic potential of Vibrio fischeri. Why don't you see dark mutants in the light organ? I mean, you'd be a very clever bacterium if you could cheat like that. Well, one of the interesting things is that the light organ has all the components of the visual transduction cascade and can see potential cheaters. But we recently looked with an M-Cherry GFP construct again, and actually there are some cheaters in there. There are some dark individuals, and this is with being colonized by ES114. In other words, a clonal population is not, all doing the same thing. There are some cheaters within there, and now one of Ned's former postdocs just took Tim Miyashiro just took a position at Penn, and he's looking at what are those individuals doing. But we have lots and lots and lots of strains that we use. The one we typically use is ES114, but we haven't seen any difference.
And I'm going to ask an even more basic but clinical question. You said there are 10 organ systems that interact with the microbiome. I mean, I can think of 4 or 5. I'm not sure.
Oh, are you going to? I feel like I'm in my oral exam. So, so there's integumentary. So the vertebrate biologists help me. Integumentary, reproductive, respiratory, endocrine, nervous. Did I say reproductive? I said reproductive. Excretory. Did I say excretory? I need to. I need to know what I've said already.
That's great.
But there are 10 vertebrate organ systems. Musculoskeletal. Give me another chance. No.
If there aren't further further questions? If there are, we can take them. If not, there's a reception across the hall in the library. You're all welcome to come and continue discussion with Margaret or each other.
Thank you.